Hello everyone, thank you for being here, and today I'll be taking you on a journey. This journey looks inside the plant leaves with the help of microscopy and artificial intelligence. So let's go and see the... the title of the talk is Inside Plant Leaves, and we'll be looking at two major concepts, that's microscopy and artificial intelligence. And to begin with, finally, I'll take you to nature, where we see plants. You see them every day. You see flowering plants. You see these huge, humongous trees in nature. And you also see these tiny little growing plants on the rocks and pebbles and stairs and moist conditions, just this thing. And so the most common thing with every plant that you see in nature is the plant leaves. There are big, small, different sizes and shapes all together in nature. And this is what we really study in the lab, the plant leaves. And to do that, we use this plant, which is known as Arabidopsis thaliana. So this plant is the most favorite of every plant scientist that is working in the world, and it looks like these tiny plants. So these tiny plants are actually solving the world crisis of crop production, which I'll show how we're doing that. But inside these plant leaves, we have a structure, a 2D structure. This structure comprises of the epidermis layers, which are like the boundaries, and protecting the most important layer known as the mesophyll layer. This mesophyll layer comprises of two types, the palisade cells, which are quite rectangular, tightly packed, and we have the spongy mesophyll, which have large intercellular spaces. They are tiny little round cells. And this particular layer, the mesophyll layer, is actually responsible for the most important process happening on planet Earth. That's the photosynthesis, which produces the oxygen that we are breathing and also produces the food that we eat every day. But when you look at this structure, the 2D structure of the leaf tissue, scientists have, from many, many years, scientists have basically studied these 2D structures. These 2D structures are the structures that you see in your textbooks, in different articles, and scientists have put a lot of efforts in studying these cells individually as well. Even in our lab, we kind of try to capture the 2D images of mesophyll and study the structure and different processes that the structure does. But no one has ever attempted to look at these structures in 3 dimensions. And so this is the most interesting aspect, that we know that a structure looks like this in 2D. How does it actually look like in 3D? And well, to look at these structures, we use microscope. Well, to study the 3D structures, our lab made the first kind of an attempt to study the epidermis layer, which is the layer protecting the mesophyll layers. And you see these tiny little structures, the stomata, through which the CO2 goes inside and the oxygen is going to come outside. Scientists have also studied them in 3D, but nothing on mesophyll, which remains the most critical layer, yet we don't know how it looks in 3D. And this is the interesting aspect. But to look at it, we can use a 16th century Robert Leeuwenhoek's microscope. You can go a bit more advanced, and I'll use a little compound microscope which uses sunlight, or maybe use a bit advanced LED fitted cameras there. I use a better microscope which is quite there, and I know it's there in John Abbott, but you get to see this microscope when you do your undergrads for sure. And so all these microscopes are pretty much focus on natural light or the LED light. That's it. That's the only 2 light sources that these microscopes have. But what we use is this giant $1 million confocal microscope. And you see this huge setup of different little tiny instruments that are attached to this microscope. But the most critical thing with this microscope is that we don't use any LED or natural sunlight. We use the laser lights. And so, what's the major difference between a compound microscope and a confocal microscope? I can't tell all the concepts, but I'll show you 2 major concepts with a demonstration so that you better understand what's the difference between a $3,000 microscope versus a $150,000 something microscope. And so, a normal wide field microscope or a compound microscope, as it's say, uses a LED or a natural light, which is going to illuminate your sample over here completely. And this illuminated sample can be captured with the help of a lens or a camera, something like that. While in a confocal microscope, we have a laser, which again is going to reach the sample, but if you notice, it's not going to illuminate the whole sample, but it's going to illuminate only a single point in the sample. This happens with the use of a pinhole structure. So a simple plate with a small hole is basically $100,000 altogether. And with the help of this structure, we can actually converge the light onto a single point on the object altogether. So the first concept is converging the light with the help of lasers. The second concept is you add a drop of a liquid over your lens and over your sample. Sorry, over your light source and over your sample. So putting a liquid, having a structure and a plate and few other tweaks, the companies are going to sell $3,000 thing into a multi-million-dollar business altogether. So it happens, but the magic is that this structure, can you see anything? Pretty blurry, can't see anything. What if I put a drop of water or a liquid onto this cat and try to image it? Little better, something's there. But what if now I want to really image this cat in 3D, capture each and every tiny detail of this cat? I'll use a laser along with the liquid to kind of capture this cat. Trust me, it's not ChatGPT. I'll show you. So yeah, you can capture the whole cat in 3D and even the tiniest of the different structures with high resolution altogether. This is the power of a confocal microscope altogether. To better demonstrate that, I have this kind of a structure over here in this glass beaker. I don't think so anyone can see anything, but what if I put a liquid, which you can drink this one for sure, and as I start pouring the liquid, the object, which was quite invisible to our eyes, becomes invisible to us. And so the structure, which you cannot even capture with a laser light, you can actually start capturing that particular structure with the help of a laser light altogether. And so this is the basic, simple concept of a confocal microscope. You use a laser, you have a pinhole, and you use a little media altogether to image things. And how do we look things in 3D? So as I mentioned, that you can illuminate a point source on the sample. But what if you have another layer that you really want to capture? Well, simply move the light a bit down, and it's going to converge at a different point on the sample. Well, in a technical microscope, it's not the light source that actually moves, it's the sample that you can move up and down. And thus, this point of light can actually move across different layers. And so, if we keep a plant leaf under this microscope, you can see different layers. You can see the epidermis, which is the outermost layer. Coming to the middle layer, you have the palisade mesophyll, rectangular cells, which pretty much don't look rectangular here. And the spongy mesophyll, which was something like circular, but they are not circular. They are pretty much irregular in shape. And they are irregular in shape because this is a spongy cell in 3D. Oh, we have 3D printed structures. And so it's not circular, the way you see in the textbooks, but it's actually pretty different. And this is what regulates the process of photosynthesis altogether. But now you have captured all the layers. How do you see things in 3D? Well, for this, we use artificial intelligence and machine learning models to kind of assemble each and every single cell with a different color and different label to assemble them in 3D. So presenting is a 3D mesophyll model, first done by our lab And this particular model can be dissected into each and every single different cells altogether to be studied. Thus, with the help of a microscope and few of the complex algorithms, which trust me, I don't write, I have no knowledge of Python at all. But yeah, with the help of collaborators, we do a lot of good stuff, and we can always do that and image the mesophyll layer in 3D. But why we do this? Why we need to study a structure in 3D, put a lot of money, and what's the relation why we are studying this structure? As I mentioned, this structure is responsible for photosynthesis. The only structure in the plant that is responsible for photosynthesis. And thus, everything relates to crop productivity. So here's a map from 2015. I don't have a new data. But here's a map of 2015 which shows 2 major clusters of some of the most important cash crops from Canada. That's wheat, barley, and canola. And so these 2 clusters, the major agriculture clusters of Canada, are in a big danger due to global warming altogether. While the policy makers are working on how to stop global warming, we scientists have a responsibility to have a sustainable future for every citizen on planet Earth. And thus we need to study this structure in better ways so that we can actually understand how this structure will get affected by climate change, which ultimately is going to affect the photosynthesis process. And so when we understand this concept, we can edit the genes to have better mesophyll structures, thus having better crop productivity because we are going to have better photosynthesis. Or we can also have the bio-inspired design. So here's a prime example of Velcro designed in 1941, which actually was the idea which we got from nature, the fur ball plants that we see here. Also, with this particular study, we can have the engineers working on and predict the future. For example, your Google and iPhone can predict what's going to be the traffic at 6 p.m. on Highway 20. While we scientists can also predict, with the rise of global warming, how the mesophyll structure and how the photosynthesis is going to get affected, and which ways should we really work and do gene editing to kind of improvise the structure for better climate and better crop productivity, leading to sustainable farming. So at the end, I would like to acknowledge my lab, my supervisor, Dr. Anna Dykeman, collaborators from Yale University, a lot of funding university, funding bodies, and thank you everyone.